Cells employ a variety of signaling pathways to communicate with their neighbors and sense their environment. These pathways are important in determining whether it is appropriate to grow and divide and perform specialized functions, or if the cell should remain dormant. Two pathways which play a number of important roles in a diverse array of cell types are the Notch pathway, which is important for communicating with neighboring cells, and the mTOR pathway, which is important for sensing the levels of growth factors, nutrients, and other environmental conditions. When these pathways become hyperactive, they can lead to the development of cancers and other diseases. For instance, mutations that cause Notch to become hyperactive are the cause of over 50% of T-cell acute lymphoblastic leukemias. We know that drugs inhibiting these pathways can greatly reduce the severity of disease, but the drugs currently in use cause a number of severe side effects. Imagine that your neighbors and their friends kept parking in your driveway and you wanted to prevent this. Ideally, you'd block off the end of your driveway, preventing your neighbors from using it, but allowing them to get to their own home. This is not how the current generation of drugs works. Imagine instead that you blocked off your entire street. You've effectively prevented your neighbors from using your driveway, but you've also prevented them from getting to their own homes. This is how the current generation of drugs functions. They block early points in the pathways, which are important for regulating a number of processes in a diverse array of cell types, leading to severe, widespread side effects by preventing the pathways from functioning in cells where they have not become diseased. In the Osborne lab, I'm working to identify the specific point or points of interaction between the Notch and mTOR pathways in CD4 T cells. The hope is that if this point of interaction is unique to T cells, or at least not common to many other cell types, that it could serve as a potential therapeutic target, allowing us to inhibit these pathways in diseased cells without the widespread side effects caused by current generation of drugs.